Hello everyone. Today, we are going to explore an article from the iMeta journal. It's titled Comprehensive Multi-Tissue Epigenome Atlas in Sheep, a resource for complex traits, domestication and breeding. We know that whole genome association analysis has been widely used to identify candidate loci related to important economic traits of livestock, but most functional sites are in non-coding regions with strong linkage to sequilibrium. After the ENCODE and roadmap projects, the Fang initiative provided abundant resources for annotating farm animal genomes. However, sheep genome functional annotation lags behind other species. Therefore, we aim to build the multi-tissue epigenome atlas and explore the utility of functional elements in interpreting adaptive evolution and complex traits in sheep. The main research results are as follows. Firstly, we identified a total of 753,723 non-redundant functional elements, with over 60% being novel, and constructed the first multi-tissue epigenome atlas in sheep. Secondly, 6 chromatin states were predicted, mainly including promoters, enhancers, attack islands and repressive regions. Simultaneously, we constructed a genome-wide map of chromatin states. Additionally, we analyzed the cross-species conservation of chromatin states, and found that regulatory elements were more conserved between species with closer evolutionary relationship. To further explore the relationship between regulatory elements and tissue-specific gene expression, we identified 8,334 tissue-specific genes, and the predicted functions of these tissue-specific genes accurately reflected the specific biology processes of various tissue. Concurrently, we identified 184,019 tissue-specific enhancers, and found the genes overlapping with tissue-specific enha all exhibited known biological functions in the corresponding tissues. Next, to explore whether genomic regions, influenced by domestication and improvement, are significantly enriched in functional elements, we performed a population structure analysis, selection signal analysis and chromatin state enrichment analysis. The results showed that spleen and hypothalamic specific promoter were highly enriched in the domestication stage, while in the breeding process, the longissimus dorsi and rumen specific enhancer as well as tail fat specific promoter are highly enriched. Tail fat deposition is one of the most important economic traits of sheep and is closely related to the economic benefits. Therefore, subsequent research focused on tail deposition. Firstly, we conducted the selection signal analysis on sheep populations with distinct tail morphologies, and found that genomic regions on chromosomes 13 and the 15th may be candidate regions affecting sheep tail fat deposition. Secondly, we performed large-scale GWAS for tail fat weight traits, and narrowed down its candidate region to chromosome 13. Moreover, by integrating epigenomic data, we further pinpointed to candidate causal variants related to the deposition of tail fat in sheep. See Figure C. Finally, we counted the AL frequencies of these two SNPs in different tail morphologies, along with the tail fat weight, and the expression of nearby genes among individuals possessing different genotypes. The results found that only the change of the CH or 13517609995 a greater than C SNP had an impact on the expression of the BMP2 gene. Further verification of the BMP2 gene was carried out at the cellular level. The results indicated that the expression of BMP2 influences the differentiation of precursor adipocytes in the sheep tail, and the accumulation of triglycerides within cells. Overall, we established the first multi-tissue epigenomic blueprint of sheep, to fill a knowledge gap in existing sheep atlas datasets. This atlas provides foundational data resources for understanding the adaptive evolution and complex economic traits of sheep. Through integration analysis of our multi-layered datasets, we identified a novel causal SNP that could serve as a genetic marker for reducing tail fat deposition in sheep breeding programs. These findings provide valuable resources and molecular marker applicable to sheep breeding, helping to accelerate genetic improvement. That's all for my presentation. Thank you for your attention.